just said, Özkan Özdemir, assistant professor at the Acıbadem University Faculty of Medicine in Istanbul, and his talk will be on fair biobanking, the role of biobank directories in the discoverable and reusable biospecimen research. And the second speaker of today is Dr. Arnaud Jabouille from Inserm, University of Bordeaux, France, and he will be talking to us about protocols to practice, a methodological framework for creating and harmonizing SOPs across research communities. Dr. Özdemir, floor is yours. We are happy to hear your talk. Thank you.
Thank you. Thank you. Let me share my screen first. Just a sec. Yes, I think everybody can see now.
Yes, yes.
Thank you. Welcome, everyone. I'm Dr. Özdemir from Acıbadem University, Faculty of Medicine. Today, I will talk about fair biobanking, specifically the role of biobank directories in making biospecimen resources discoverable and reusable. Uh, so, yeah, yes. We will start by defining, today's talk will be, we will start by defining biobanks, why they matter. Then we will look at the challenges that we face with big data and so on. And the core of the talk will be about the FAIR principles and how biobank directories, especially in BBMRI ERIC, implement them. So, what are biobanks? Biobanks can be divided in two types, actually. One of them could be the data biobanks that store the multi-omic data sets, clinical data sets, imaging data sets, and so on. And the biological sample repositories that store physical specimens like blood, tissue, DNA, serum, and that kind of biological specimens. So many modern biobanks, however, are hybrid now. They maintain both data and samples together. Here you can see the photos. This is from our biobank in Acıbadem University. This is also inside a undiagnosed disease center, called Ajar. We have a rare disease biobank that we try to implement the both data storage and also as well as the biospecimen storage together. Yeah. So why? So to understand the sense of scale in BBMRI ERIC network alone, there's over 400 biobanks, more than 2,900 collections across 32 countries. This means roughly around 100 million biological samples. And these resources are essential for large-scale studies in rare disease, cancer genomics, and population studies, and so on. But this is the key point here. The value depends entirely on whether researchers can find and access to them. Without accession, this means nothing. So this is where FAIR principles and directories comes in, basically. So what are the challenges that we are facing in FAIR biobanking? It starts with data volume. It is the biggest problem, and it's growing exponentially with multi-omic technologies. These high throughput data sets are coming along, and even today, we were discussing about how to store new long-read sequencing data in our biobank because they are big, quite big, and I will talk about this later. We have to hold this with metadata, which this takes even more space than it appears. The second challenge that we face, let me take my laser pointer here, integration. It is difficult because institutions are usually using different formats about the data sets that doesn't have industrial standards. The other one is quality and standardization. This is varying widely. The metadata is often incomplete or inconsistent. This creates a lot of problems. Security and privacy, like in GDPR, PR, and in Turkey, it is KVKK, we say, adds even an additional layer of complexity to holding this data. And interoperability suffers from the lack of the shared vocabularies, basically, and institutional silos that prevent collaboration and reproducibility. And these problems collectively make it very difficult to reuse the biospecimens across institutions. That's where we need our paradigm shift. So, before FAIR principles, before FAIR principles, biobanks were isolated collections with manual catalogs and no standardized metadata or anything. But after FAIR principles, verification, we aim for networked, discoverable biobanks with machine readable, this is important part, machine readable directories, and rich and standardized metadata. The shift from isolated silos to connected searchable networks is what makes collaborative biomedical research possible at scale. Okay, so what is this FAIR principles? It is published, the principles were published by Wilkinson et al. In Scientific Data in 2016. These are these principles are not defining any standard. It rather gives a set of guiding principles for scientific data management, how we manage this data. And what makes it unique is the emphasis on both human and machine readability. This is the important part about FAIR principles. That's why we are just shouting about it for 10 years now. So let's dive into the first findability, the F of FAIR principle. The data in findability, data should be assigned a globally unique and persistent identifier, should be described with rich metadata. Metadata created from the the initial data should be clearly and explicitly include the identifier of the data. And metadata should register and or indexed in searchable resources. This is exactly what biobank directories do, by the way. In the biobanking context, this means DOIs like for sample collections, schema.org and bioschemas markups for search engine indexing, and registration in directories like BBMRI ERIC, making it machine readable by using APIs and JSON formats, as in here, is make this how this biobank directories make verified. When we dive into accessibility, this is once the data is found, it should be retrievable with standardized protocols. This is the important part. How we do that is data should be retrievable by their identifier using standardized protocols. The protocols, protocols that we use should be open, free, and universally implementable. The protocol should allows authentication and authorization where needed. And metadata should be accessible even when you don't have the data even. These three here, you see the three steps access model: registration, authentication, and authorization is how, again, BBMRI ERIC implements this principle as biobank directories. When we dive into interoperability, data should use a formal, accessible, and shared and broadly applicable language, should use vocabularies that follow FAIR principles, and also should include qualified references to other data. So in biobanking, there's key standards include MyAbyss. The minimum MyAbyss, the longer version is minimum information about biobank data sharing standard. The FHIR and AL7 for clinical data exchange. SNOMED, CT, ICD, or HPO even for clinical annotations. And for documenting how samples were collected and processed, we use this sample pre-analytical code here, SPRAC. So those are the shared interoperable protocols that everyone can use freely. Those are freely available. And when we dive into reusability, the ultimate goal here is optimizing data to reuse in new research contexts. So data should be richly described, should be released with a clear, accessible data usage license. Licensing is really, really important, by the way. Even though it's open license, without license, user cannot understand how and why, how this data could be used, basically. And the data should associated with detailed provenance information and meet domain relevant community standards. And for biobanks, this means clear consent terms, material transfer agreements, detailed sample provenance and tracking, Creative Commons licensing for metadata that we create from the original data, and quality indicators such as pre-analytical variable documentation via SPRAC. Yeah. So this is our slogan: as open as possible, as close as necessary. This captures the essence of FAIR principles, FAIR biobanking. We want to promote reusability, accelerate research, while also protecting the confidentiality of people and subjects. This is where FAIR plays a quite big role. So let's talk about practical infrastructure that makes fair biobanking possible now. In biobank directories, that are centralized searchable catalogs, basically, that aggregate metadata about biobanks, their collections, and available samples. They serve three key functions. One of them is discovery. Researchers can search by disease, material type, country, or sample size. The other one is connection, linking researchers with biobank managers through standardized request systems. And the other one is assessment, evaluating sample quality, collection size, and assess conditions. The major examples could be, again, BBMRI ERIC here, or ISPER, or UK Biobank Portal, or Biobank Japan. There are great examples like these. Here you can see the BBMRI ERIC directory interface. As I told you earlier, this catalog has over 400 biobanks with more than 2,900 collections across 33 countries. Here you can see Acıbadem Biobank is also listed here as a BBMRI node located in Turkey. We are one of the four nodes. The head node is IBG Biobank in Izmir. Neşe Hoca here is the director of it. And we have Istanbul University Biobank, and also we have Hacettepe Biobank, other than our Acıbadem Biobank. So BBMRI ERIC is a federated platform that goes beyond the directory, actually. It is a four-step pipeline, basically. First, here, it is a directory that provides the central metadata catalog for browsing. Second, the locator and finder enable privacy-preserving federated queries. You can build virtual cohorts without moving sensitive data, basically. Third, negotiator. This is really great. That streamlines the access negotiation between researchers and over 600 biobanks. It facilitates MTAs and communication and so on, that kind of stuff. This is quite useful. And also, The fourth part here is controlled access and reuse under GDPR or KVKK in Turkey compliance. Uh, Turkey is now an observer member of BBMRI ERIC, and basically we are a part of it for a while now. Yes, yes. So this slide maps four fair principles here and how it is used in biobank directories. For findability, for example, here, persistent IDs could be used with rich metadata, search engine indexing with bioschemas. This is for findability. About accessibility, HTTPS protocols are also standardized APIs, and authentication for restricted areas creates this verified accessibility protocols. In terms of interoperability, JSON files markup, MyAbyss standard, as I told you, or cross-link links to the Elixir ecosystem, which I will talk later on, including this RDM kit and fair sharing org and bio tools. As you can see here, and reusability for the reusability, this CC BY for licensing or credit attribution ontology or ORCID for authors. These kind of protocols make fair biobanking available. Here you can see the fair implementation tools in practice. The technical standards and platforms that connect biobanks to broader FAIR ecosystem. And these include schema.org, bioschemas, MyAbyss, as I told you, and various Elixir sources. In Turkey, we create a platform called fairveri.com. Basically, what we provide here is guidance and tutorials in Turkish for implementing these tools in your own biobank or research, basically how you hold scientific data. It's about that. Elixir Europe is an intergovernmental infrastructure of life science data. This connects 22 national nodes plus the EMBL hub. And their core fair resource are invaluable for biobanking, like BioTools is registry for bioinformatic tools, fair sharing catalogs, standards and policies. This RDM kit provides data management best practices. Tests is a training portal for life sciences. Uh, uh, yeah, Fair Cookbook gives practical recipes about how you can verify your data. And Bioimage Archive is basically a reference image data repository. And that this data stewardship wizard is great, again, for fair data management planning, how you define your biobanks, basically. So this is where ethical and legal frameworks has to be told. uh, Ethics in biobanking rest on three pillars, basically, as I mentioned here. Uh, it's about informed consent forms. We need informed consent forms, dynamic consent models, and must choose between broad or specific consent depending on our biobank's positioning, while respecting the right of withdrawal and regulations, by the way, withdrawal. In Europe, we have GDPR. In Turkey, we have KVKK law, which covers special category personal data and requires data protection, impact assessments here. And about governance, institutional review boards and data access committees are mandatory, and basically material transfer agreements or ethical advisory boards are essential to make this biobanks verified. So if we talk about the biobanking in fair biobanking in Turkey, uh, for findability, uh, Enable's platform is a great example that centralized the health data, but international metadata standardization is still limited. For accessibility, citizens of Turkey Republic can have free, secure access to this platform, but research accession and technological literacy needs improvement. For interoperability, national health institutions are integrating both alignment with international systems like FHIR, but still it is incomplete. For reusability purposes, personal health data reuse is enabled in some contexts, but data licensing and anonymization has still needs further development. The key stakeholders here are TÜSEP, Ministry of Industry, Ministry of Health, academic institutions, and Turkish Fair Resources basically are the key stakeholders. It needs still development. However, our international collaborations allow us to integrate many things together. So here are the five key challenges and solution pairs, as you can see here. Uh, Lacking of standardized metadata across biobanks. This is the most maybe the common problem that challenge that we have in data biobanking. What we need to is adapting to MyAbyss 3.0 and bioschemas. About privacy and data sharing, we need to use dynamic consents, federated learning, and differential privacy. Now there's this new kind of tools that we can use for each of them. And also about this institutional silos, intensive sharing through citation credits and funding requirements. These technical barriers to interoperability, we need to use shared standards and APIs. And about sustainability of biobank directories, long-term funding models and national biobank strategies are mandatory. As for future directions, we need four major directions that will shape fair biobanking. Well, the first one is federated learning, which allow us training AI models on distributed networks of biobank data without moving sensitive records. Second, the dynamic consent platforms let donors manage their consent in real time. AI-powered metadata enrichment can automatically tag and link biospecimen metadata using NLPs. And national biobank strategies, as I told, can connect country-level directories to international networks like BBMRI ERIC. Turkey has a significant opportunity here, I should mention. As takeaways, first, FAIR principles provide the framework for discoverable, reusable biospecimen resources. Second, biobank directories are critical infrastructures that are front door to biospecimen research. Standardization is important. Ethical and legal compliance through GDPR or KVKK with dynamic consents must be a new goal for verification of biobanks. Fair is a journey, not a destination. This is another slogan of fair data, which means continuous improvement, training, and community collaboration are essential here. As my last slide, I want to told you about fairware.com, which is a uh, which is a platform that we created for fair principles knowledge base. This is a Turkish language resource hub for fair data. You can basically dive into that platform and getting know-how about fair, how your data could be more reliable to fair principles. And also you can have some tests, you can download some verified data. There are some questionnaires and that kind of stuff basically is in the fairware.com. Uh, Thank you for this opportunity.
Thank you, Özkan hocam. If you can stay with us, that will be great.
Yeah.
Until the end of Arnold's talk, and we will
Mhm.
get the questions all together. Thank you so much. So we can move on to next speaker, Dr. Arnaud
Okay, yes.
Jabouille.
Let me share my screen first.
Thank you.
Okay, you see my screen?
Yes.
Okay, so all right, so let me introduce myself. So my name is Arnaud Jabouille. I'm a researcher in the Bordeaux Institute of Oncology, and today I'm going to present you the structure framework we developed in Working Group 2 to move from individual laboratory protocols to harmonized transferable SOPs across the different research communities. So sorry. Okay, so my talk will be organized in 4 different parts. First, we will see why harmonized SOPs matter. Then I will walk you through the methodological framework of building and harmonizing SOP. And then I will have a short focus on reviewing and the acceptance process of these SOPs. And finally, I will have some words on how AI tools can help you harmonizing the SOPs that you develop. So first, why harmonization matters. So the main challenge is the multi-site challenge, because modern biomedical research increasingly relies on multi-institutional collaboration. In large collaborative research networks, such as the Translacore, the scientific innovation often progresses faster than the procedural harmonization. And in our community, we can see that our community is represented by multiple institutions that are spread across more than 20 countries. And this diversity generates a lot of heterogeneity. First, heterogeneity in lab practices and practices and also protocols, because each lab operates within their own technical culture, but also historical practices and regulatory environment. This regulatory environment depends on the legislation relevant for biomedical research. And this legislation is dictated by the European Union that will direct and edit regulation and directives that will be transposed by the different countries into national laws. But on the top of that, the countries also add their own additional laws that will create a very divergent frame for the regulation of the biomedical research across the different countries and institutions. Another cause of heterogeneity comes also from the pre-clinical handling. Due to the heterogeneity and the lab practices, even small differences in sample handling can have and introduce a significant variability in omics and datasets. So, and then when the data are later compared or integrated across a site, this hidden variability becomes a major obstacle to the reproducibility across the institution. So this reality even matters more in translatomics and omics research, because in omics, We're not simply measuring stable molecules, but we are capturing dynamic biological states. And these dynamic biological states are characterized by different specific parameters that are extremely sensitive to how the samples are handled before the analysis. Among these parameters, we have the RNA integrity, but also the microbiome composition, the ribosome occupancy, how the chemical modification on the genome and the transcriptome can be affected by how the handle is processed. So in translatomics, actually, this sensitivity is even more pronounced because we know that the translational programs are designed to rapidly respond to stress cues. And for example, we know that the cap-dependent canonical translation is normally shut down when some stress is activated, leading to activation of non-canonical translational mechanisms. So therefore, the pre-analytical harmonization is mandatory. It's not an administrative requirement. It becomes a biological requirement. So this is something that we have tried to implement in the working group 2 by developing standardized operating procedures. So what is a SOP? So SOP is a structure and detail document that describes the standardized processes to help workers and workers to carry out routine operation. It's designed to ensure the consistency, quality control, and reproducibility across the institution. It also defines the responsibilities, the materials, the procedure, and quality checkpoints that must be validated. And they also serve as a reference framework across the operators and institution. So in working group 2, developing such documents was for us an answer to our main question, which was how do we harmonize the pre-analytical procedures of biospecimen processing to ensure a reliable and comparable multi-site omics data. So achieving this required to integrate multiple data, sample types, and technical environments that are generated from different institutions, but in one goal to be implementable across institutions and countries. So then, how do we build the SOPs? What is the methodological framework? So for that, the first step is you need to identify and mobilize the expertise. So to do this, you first identify who holds this relevant expertise. And this is performed through a structured exploration of multiple channels, including the organizational structure and members directory of your consortium. But it also you need to also identify the biobank managers and the core facilities leaders from the participating institution, because these actors often have a deep operational expertise in sample handling, but also quality control and regulatory compliance. So then the third point, the representation, it also matters because to answer your legitimacy and the broad adoption of your SOPs, the expert group should reflect a balanced representation of all the countries composing the consortium, because what we need to reach is the harmonization of all the practices, and this harmonization must emerge from a collective expertise. So then you need to contact the relevant experts. So you can use different approaches to do that, either by direct contacting them through a targeted email, but you can also use another approach, more structures using a survey that will also help you collecting and standardized methodological information. An example of that is this table. You can see that these are the information we collected from the survey that we used in the Translacore. And you can see you can already have access to different kind of information, including name, also affiliations, but scientific expertise, scientific background. But also you will see how these experts want to contribute to the establishment of this SOP. Some will share specific protocols, but others will also be more interested in being a part in the board of the reviewing process. So the step 2, once you have mapped this expertise, you need to collect the SOPs. So you collect this SOP, but you don't immediately standardize them. You have to first map the landscape of existing practices. So for that, you map them by identifying the common practices across the laboratories. You then detect the methodological divergences between the lab, and you distinguish which parameters are critical and which parameters are flexibles. At this stage, you will be able to identify convergence points where all the laboratories practices already align, and divergent points when the procedure differ significantly. So this mapping phase actually is very essential because the harmonization must be evidence-based, and it's also biologically driven. It's not arbitrary. So the objective of SOP is the reproducibility and not the uniformity. Sorry. So once the existing practices have been mapped, the next step is to design an harmonized template that will be used to structure all these SOPs. So this standardized structures is organized in different sections. First, objective, scope, general information, safety requirements, material equipments, procedures, workflow notes. So this section is you will see is very important. You also have to reference the quality check steps and the process that you acquire the metadata. And finally, since the SOPs are intended to be integrated in a broader work of a scientific community, it should include also links to other SOPs or protocol from the community. So now, what are the purpose of each section? So the objective, clearly, the purpose establish the purpose of the SOP. It defines what the biological material will be processed and what are the potential downstream analytical application that will be used. So then you have the scope. So the scope specifies who should use the SOP and in which context. So in the context, for example, of a consortium like the Translacore, this section clarifies that the SOP is intended for specific partners involved in the network and performing multi-site analysis. So then the general information section provides the biological context that is necessary to understand why specific precautions are required. For example, in translatomics and RNA, the RNA and ribosome are highly sensitive to degradation and cellular stress. So hold the time to processing and temperature control is very important or critical parameters. So this context has to appear here. So then you have the safety section. So this section describes all the biosafety requirements that are associated to the sample type and to make sure that this sample type will be processed in adequate safety for the operators. Then you have the material and equipment. So this material equipment specifies the regions, but in harmonizing the SOP, your objective is not to impose specific brands because the SOP defines the quality standards that the laboratory must meet. So then the section that follows is the procedure. So the procedure is the core of the SOP because it's a step-by-step description of the workflow. including all the metadata collection process with all the key parameters that need to be documented across the different institutions. So these parameters are multiple. There are some that are listed here, but they're not exclusive to this one. And these parameters are critical for the traceability, and they will, at the end, facilitate the cross-data interpretation. So then you have the workflow notes. So this section is important because different downstream analyses have sometimes distinct requirements. So this section highlights this consideration. For example, for ribosec, you need to rapidly process the sample to preserve the ribosome positioning. So it's not applicable to all type of samples. And so this section will clarify this context. So then you have the section that will be linked to the other resources of the community, because SOP should not be considered as isolated documents. They should be integrated into a broader methodological ecosystem. So in practice, this means the SOP should include references to and links to complementary protocols and resources developed by the research community. And these links are essential because they will ensure the continuity between the different stages of the workflow, from the sample collection to the pre-analytical handling to the downstream analytical procedure. So to conclude on this step, the harmonized SOP template should function as interconnected resources rather than isolated document. And by following this approach, your SOP will promote the interoperability between the institution and facilitate also the adoption of harmonized practices and protocols across the laboratories of the consortium. So once you have wrote your harmonized template, you need to focus on defining the clear boundaries of this SOP. So to that, you need to answer a critical question: what the SOP will cover, and of course what it will not. And for that, you need to think based by following key parameters. And the first one is the biological logic. You need to make sure that you know the sample type you're going to analyze, and you need to define its composition. From that, you will be able to establish the molecular vulnerabilities of your sample type and derive specific procedural constraints that will dictate how you will handle and process the samples, and what are the compatible application and analysis you can apply to them. So of course, this harmonized SOP should not try to cover every possible experimental situation. Actually, it's not possible. And equally important, this SOP must explicite what are the applications that are not compatible with the described procedure. So defining these boundaries will prevent then the misinterpretation and ensure that this SOP remain scientifically robust. So let's try, for example, let's try on some examples. For example, you want to write a SOP on ribosec and epitranscriptomics using a sample type that is called the bronchial lavage fluid. So the bronchial lavage fluid is a sample that is obtained from the injection of PBS in lower airways and using an endoscope. And so this sample type is characterized by a low biomass and a high RNase activity. So clearly it's not well suited for processing for ribosec and epitranscriptomic that require a lot of material. So your SOP will mostly exclude this sample type for a reason of robustness, but it can also focus on the pre-analytical part and then refer to the analytical SOP of the community if this one is existing. Another example is, for example, you have patient-derived organoids and you want to set up and write a SOP for ribosec. So these samples are structures that are highly sensitive to mechanical stress. And as you know, the stress is a very critical parameters in ribosec. So you will add specific notes in the SOP regarding the critical processing of this specific sample and for the ribosec experiment. So when you have finished this, you need to make sure that your SOPs are aligned with the international standards. So these standards are defined by the international organization. So the international organization is composed of 176 members, one per country, and they have developed more than 26,000 international standards that cover all the aspects of the technology. So for the one that are interesting for our kind of fields, there are multiple ISO standards dedicated to biobanking and quality management, pre-examination processes for different type of samples, going from frozen tissue to RNA from whole blood, but also the way to prepare the samples for microbiome analysis. And of course, you have also ISO standard for the quality and the competence requirements for medical laboratories, but also for medical laboratories that have very high quality standards. So then the next step is the reviewing and acceptance process. So once you have your harmonized SOP, if you go back to the workflow here of the harmonized SOP development, you will see that the reviewing process is one of the significant part of the job because the first version will be then transmitted to the firstly identify experts that come from the initial expertise mapping step. These experts will give you some reviews that you need to integrate. So you need to integrate their feedback, but you also have to resolve the methodological differences that have been identified to produce a revised version of the SOP. So this revised version will be then tested and validated across different identified labs before the final approval. So the final approval is normally given by the working group leadership to then be disseminated across the community, so the different consortium channels that has been defined and produced. So as a final part of my talk, I will go to just say some words on the AI tools to support the SOP harmonization. So the first thing that we need to remind us is the use of AI tools follow and must comply institutional and national regulation. I'm sure it's the case for most of the institution. It's also the case for mine. Institutions generally have issued different guidelines regarding the use of AI tools, particularly when sensitive or unpublished research data information is involved. And it's definitely the case for SOPs, for protocols that you will be access to, because you will get different kind of protocols, perhaps also protocols that are not published. Some also you will potentially also have access to different kind of technologies, proprietary technologies. So you need to use to follow the guidelines of your institution in terms of AI support for SOP harmonization. So some of these guidelines include, for example, a training just to understand the implication of using AI. You have to use it sourcefully with adapted with a use that is adapted to your current needs. And then, for example, an interesting point is my institution will not fund paid subscription to non-sovereign external generative AI platforms. So they want to maintain the proprietary of the data. And so for that, they actually develop their own AI platform. So just to summarize on the AI, so the AI tools can support SOP development without compromising sensitive data when they used appropriately. So what is acceptable, for example, is structuring SOP templates. So you can use AI to do that. You can also use AI tools for harmonizing the terminology across the documents, improving the clarity and the readability, and also assisting you in drafting the generic methodological description of the SOP. What you are not allowed to do is to upload and provide some confidential information to the AI tools, specifically the institutional protocols and hold the protocols or the research data that include the proprietary technologies. So to finish, take-home message is the following. So we developing harmonized SOP for the research community. That's we've done in the working group too. And this required to first map the expertise across the scientific community. You need to use the biological rationale to define what are the procedural constraints, define a shared and structured SOP template that must be aligned with international quality standards. And then you have to think about how the reviewing and the validation process must be undergone. And so for that, you have to have a clear and transparent process for this step. So finally, harmonized SOP, if you follow this kind of procedures, harmonized SOP can transform the individual laboratory practices into shared, reproducible standards, enabling the reliable multi-site research. So as a last slide in working group 2, I can share with you some resources that we have developed. So we have developed harmonized SOPs for biobanking and pre-analytical processing of biological samples for omics research. So from that, we have, I think we will release two different sets of harmonized SOP. The first set will be released in the next couple of weeks. It will be focused on frozen tissue, FFP tissue, blood and blood derivative, but also patient-derived organoid, bronchial lavage fluids for DNA and RNA-related omics. After the Bologna meeting, we will try to release a second set of harmonized SOP that are mostly focused on proteomics. By doing this work, we have also generated different resources that will be available also, I think, from the website. We have generated a structure SOP template, and we also have both different ISO standards that are related to the biological fields and that which will be also available. So I would like to thank you, all the working group contributors, as well as the partner labs who shared their protocols and their methodologies, including also, wow, that's funny, including also the other working group collaborators. Thank you for your attention.
Thank you very much, Arnaud and Özkan, both of you. It was great. So we have time to get some questions from the audience.
Yeah, go ahead. Yeah, let's see if anybody has any questions. I will have one mostly to Özkan. What do you think is the biggest problem? Actually, it might be to both of you. The biggest problem when you try to create a biobank. I mean, I could tell you a little of our side of the story.
According to me, maybe Arnold has some other ideas about this, but to understand where to start is the most complicated thing,
Yeah.
according to me.
Yeah.
Where you should start is the. I'm a bioinformatician. I mostly work on the data sets and so on. That's why creating the data part of the biobank, for me, the initiation process of how I get involved in this biobank business. However, for a biological person, probably it starts with biospecimens and so on.
Yeah.
Where to start is the most important key thing.
Yeah.
A few years ago, I was helping some of my colleagues. They wanted to make something they call live cell biobanks. So they were like taking tumors, digest them, so they could collect the cells and then freeze the cells alive, so you could take them out and use them. And the main problem were clinicians.
Yeah.
They just didn't want they took these samples as private, and it was a continuous battle with them. So I don't know how you guys solve it.
Well, we have not solved it actually from our side. We're still struggling every day to get the samples. It's like an administrative battle all the time, you know.
Yeah.
But yeah, I agree. The clinician is the main barrier, and mostly also, I mean, the pathologists. You can have pathologists that are really helpful, but once the pathologist access to a sample, sometimes it consider it has their own property, you know. So sometimes it's kind of difficult to get all these samples.
Yeah.
For us, our side, for clinicians, we are in the same page. They don't understand even the concept of biobanking, how we can open the data or open the biospecimen to third parties and so on. This concept even is hard to understand. What we do to solve this in Acıbadem hospital group, so it is good that we have a private hospital group that we can easy to reach. So basically what we do is just go and have a meeting with them. This is how a biobank works. This is how easy that your resources could be.
We do the same. I think the key is really to integrate them at the beginning of the process of your project. I think this is the key. Because when you have a project that is already ongoing and then you want to validate some stuff with like clinical samples, it become already the mess. But if you have started by implementing them at the part of thinking of building the project, I think it's easier to get access to them.
Maybe the best thing is have a target clinicians or target group. Start from there, have a success story, and people will come.
Yeah, I agree.
Yeah, that's a good point. Yeah. Anyway, really nice work, because I think this is extremely important. This standardization is also specific because, you know, each department, each lab, each hospital, they treat samples completely differently. And, you know, we, again, many years ago, that was in Finland, we were trying to get RNA data from fixed tissues. And the problem is when you were checking the samples, they were treated with completely different
depends on the
preservation
fixation
methods. So,
exactly.
So
so that's why it's really important to harmonize the procedures at the beginning. Between
otherwise the data is lost, you know.
Otherwise it's not exploitable at all. Totally.
So it's a loss for everybody in that sense. So anyway, excellent work. Thank you for this. Any other question?
I have one question. So, regarding, let's say there is a big retrospective collection of FFP blocks sitting in the biobank somewhere. So I always wonder if we want to design a new study, how is it possible to get a consent from the owners or donors of these samples? How does it work? This is something I'm always curious about.
Okay, so I can answer for this because I've been faced with this kind of situation. So actually, for like our current project, we have a bank of FFP tissues that we exploit, and for which they ask us to have, because these were some were old samples, some were current clinic samples, and for the old one, they ask us to re-demand the authorization to the patient. So we had to send email to the patient and get contact to the patient to validate their approval for the new study. So yeah,
I have a
otherwise
question.
you can only use the one from the deceased patient.
Okay,
So
if they are deceased, no need to.
Yeah, if they are deceased, you don't need the approval. You can do whatever you want with the sample. But if they are not deceased, and for example, sometimes you also have differences because now we have implemented, for example, in our hospital, one-shot process. Once the patient get like an exam, he already have a notarization document to sign. So it's included in the clinical routine of the patient. But if it's not the case, and because it's also a new study, you have to go through contacting again all the patients and get the approval for the new study you want. I mean, this is what we have to do for our study, actually.
May I make a comment?
Of course, hocam.
Actually, if patient was deceased, also you have to contact with their relatives. But everything depends on the consent, actually. If you have a consent of reuse of these samples for different studies, you can keep using that samples. But if
Okay.
you don't have any consent of that patient, you should get approval from patients or relatives. That's why biobanks are really important, because when you have a
Yeah, you already have
biobank
the consent
it means that you have rights to reuse those samples for different studies, and also you have rights to share those samples.
Yeah.
That's why biobanks are really important. And I'd like to, as a working group to work package leader, I'd like to thank you all for your participation, and also for the experience that you shared, because the scope of this work package actually is first determine how we can access already established cohorts for our research. Özkan already mentioned about the biobank directories and how we can reach them. And also our idea is to include their actually web page and addresses in our Translacore web page, so you can easily create your cohorts with that already established biobank samples. And the second aim is also sharing our expertise and harmonized procedures with each other, and to boost actually collaboration between Translacore partners. So your presentation, Arnaud, was really helpful for actually how we can use harmonized procedures for actually long-lasting collaborations and creating really really valuable data at the end of our research. Yeah, thanks all. And also thank you for this opportunity.
Yeah, thanks a lot.
Thank you for volunteering. Theo wanted to ask a question, not anymore.
Yeah, but I'm not sure. Hello, everyone. I assume that all these data are collected usually through a clinical study. Is that correct? And they stored at the biobank. So, what is the problem of if they are going, let's say, to stored, I mean, the samples, and then the data is anonymized, then can be accessible to the rest of the community. Am I missing something? What is the problem over there? And discussing about how to engage clinicians and things like that. If they are part of the clinical study, they are contribute to the study.
Yeah, yeah, yeah.
I don't know if I missed something.
No,
What is
I
the
agree. I
problem that
think,
you actually
I mean, for our point of view, sometimes because clinician, I mean, if normally it's, so for example, in France, you have the Inserm that normally drive the research in the health, research in health, and you also have hospitals that mostly do clinical trials, okay? And sometimes their vision are different. And so it's kind of a political issue. But I think the key is really to, I think we convened, is to really implicate the clinicians at the beginning of the study, and that way I think you disintricate the whole complication that you will then have later on.
I think the problem is this. You can collect biological samples for project-based manner, or you can collect them as biobanks. If you collect them for a project or for clinical diagnosis, you may not have actually chance to share those samples with others.
Yeah.
It depends on the consent. So if a sample was biobanked, so it is good, you can use them for further research, but it is collected for a specific kind of research or specific project. You should actually remove all of those samples at the end of that project.
One other thing about genomic data, it is basically you cannot anonymize it because it is already unique and related to one person by itself. That's why we are created a term called pseudo-anonymization, which is basically you separate the
Özkan,
name and
pseudo-anonymization
yeah,
is
yeah,
the
separate
step
the name.
before the bread, but can be fully anonymized. But there are certain ethical issues in rare diseases and things like that. For observational data, for some clinical and things like
Yeah,
that,
those kind
it
of
is,
data,
and this
you're
is
right.
Europe goes in this direction. And one critical issue is how all this data eventually goes to electronic health data space that we aim to develop at a European level. So the research community must support this movement in order to have all the wealth of the federated, let's say, accessible data for the research community. But I'm not sure that we are there. We need to do a lot of things anyway.
Actually,
So,
technologically, we are there because we already have a federated infrastructure already built up. Just we
of
need
course,
is standardized protocols and give more
that we don't
information
have
about how to share
verified
data and so
data.
on. What? I'm sorry.
We don't we don't have a lot of verified data. I mean, it's so hard and difficult to verify them. Anyway, we
This
will
is
go there
correct.
eventually.
However, AI is getting great, and if we are able to build up strong federated networks for our studies, we can just use this federated learning stuff to
Of course.
create great metadata and so on, basically, from out of this research. Mhm.
A nice presentation, Özkan.
Thank you.
I think there is one question in the chat. Arnaud, can you read it?
Okay, so actually we are still working on. We've received the SOPs to harmonize, so that's why it will be not a part of the first set of harmonized SOP that will be released through the working group 2. But then definitely they will be released, so you will be able to reach this SOP for sample processing for proteomics. So we're still working on it, but after the Bologna meeting, normally this kind of SOP should be released.
Okay. If Theo is asking a question again.
Do we have the time?
Sure, go ahead.
Okay, I'm not sure what are the developments to the different working groups, but the SOPs at some point will be harmonized, hopefully, and we are going to come up with some recommendation. Are we aiming to have, let's say, to the regulators or to EMA, I don't know who is going to be the main recipient of this, the key stakeholder. So this going to be disseminated somehow, and the whole community, because it's not obligatory. Yeah? So I'm asking this because this is a kind of, let's say, exercise of impact, creating impact out of this cost action, how this, the outcome of this wonderful work can found the way to the clinicians, everyone is engaged in this field. Thank you.
So I don't know if I can answer, but I think first for the SOP, so there will be a first set of SOP, I think, available from the website. But then another approach has been also, I think, engaged from Denis, who is to set up a chapter book to detail how these harmonized protocols to study translatomics. And so this is actually ongoing. And this will be, I think, probably finalized before the end of
GLBs.
Sorry?
Are we going to develop some GLB, good laboratory practices? This is what we aim
I think
to do.
it comes from the specific identified experts for each type of analysis. I think at least for the book chapter.
What I'm just trying to say is that let's think the next step, how this, let's say, the outcome of this work will find its way to the research community, to the clinicians or regulators. I don't know what we are going to eventually, create impact.
I think you will need some people at the very top level of the European Union to harmonize, I think, some procedures. I don't know.
Yeah, I think it will be discussed in the next management committee meeting. What would be the further progresses on Translacore?
Yeah.
This is beyond us, I believe.
Yeah, I believe so too. So if there is no further questions, so I thanks first the speakers, Ahmet, Özkan, and Arnaud for the really nice talk and their work, of course, and everybody else who has been patiently listening to them today. And we will have the next webinar next month. We will send you an announcement as soon as we can.
Okay.
Yeah. So thank you once more.
Thank you so much.
Thank
Bye-bye.
you very much.